this is how it was named. Now, coming to the MHC complex introduction, what it is. So basically, this is the genetic factor, and this reside on the chromosome number 6, on short arm of chromosome 6, where the class 1 MHC. This MHC complex is divided into 3 types basically, and class 1, 2, and 3. The class 1 reside towards the telomeric end, and class 2 resides towards the centromeric end. And in between these two, there is class 3, so which is primarily associated with inflammatory responses. And these class 1 MHC complex are found on all the nucleated cells, and class 2 are basically found in hematopoietic, 3 hematopoietic cells like these lymphocyte, dendritic cells, and the macrophages. So now what is significant in this, that what happens at the cellular level. So this is the picture when crystallographic picture has shown it's a 3-dimensional picture, but this cartoon is showing the 2-dimensional picture and its structure. If you can notice, there is class 1 on the left side, there is the class 2 on the right side. The class 2 is basically, there are 3 alpha chain, like alpha 1, 2, and 3. They bound with beta 2 microglobulin, which anchor it, and with the cytoplasmic tail. Beta 2 microglobulin resides on chromosome 15. Now, what is most important over here is the groove, antigenic groove, where the peptides are bound, that is formed in between alpha 1 and alpha 2. Now, compared to alpha 1, MHC class 1, class 2 is a heterodimer structure. So it is formed by alpha 1, alpha 2, and beta 1, beta 2. And the peptide binding groove is like in between alpha 1 and beta 1. And similarly, it has got cytoplasmic tail. Now coming to exon intron organization of the class gene, these exon encodes different structure, like exon 1, they encode leaded peptide. So leaded peptide, and 2 and 3 exon is the most important in terms of immune system, because we determine this HLA typing of alpha 1, alpha 2, which reside on the which encode the peptide binding region. Now exon 4, it encodes alpha 3, and similarly exon 5, 6 has the different identity. So what we try to find out is the peptide which are binding between alpha 1 and alpha 2, and in MHC complex 1. And similarly, the MHC complex 2, where there are alpha 1, alpha 2, and beta 1, beta 2 are there. So here the group antigen peptide binds in the group of alpha 1, beta 1. So we try to find these genes over here. Now, what is most important, this is the genetic part. Now, what is most significant, how these cellular events take place inside this, inside the cell. Now, these MHC class 1, which reside, they have endogenous pathway. Now, the class 1 MHC molecule, when they have to process these endogenous peptide, what they did do that they combine with the calrexin and a bunch of basically chaperone, calrexin and ERP57. Now this calrexin behaves just like a mother who has to make this class 1 with the peptide binding site. So now they form a complex, they are associated, and she remain over there, rather calrexin remain over there till beta 2 microglobulin bind with them. And then after release of this calrexin, tapacin and calreticulin binds with them, and they form a complex structure. And in the meantime, the protein, endogenous protein, which has to be, which is processed with the proteogene, they migrate with the help of tap protein, and they migrate inside the endoplasmic reticulum, and then they are processed. So this complex, unless this ERAP will not find a suitable antigen, they are not going to release. So ERAPs basically it disintegrate this and produce a protein of definite size which it binds. Once this bind, and then the whole complex they are released, and this is how the class 1 MHC molecule present the peptide on the surface to the CD8 cell. Now, this was the class 1. Now, coming to class 2 process exogenous pathway. Now, class 1 endogenous pathway and the class 2 exogenous. So, what happened? These exogenous protein, which are outside, they are in phagocytosed inside the cell, and they have to combine with MHC, whereas the MHC, newly formed MHC, they are their groove are with their group is occupied with invariant chain, and until they are digested they remain over there in the form of clip. Now this clip has to replace with the exact peptide which is helped by HLA-DM mediated. So HLA-DM mediates the clip for antigen, exchange of clip for the antigen expressions. Once this exchange takes place and the exact peptide binds with this, and then they are presenting this peptide to the surface, and then finally they goes to CD4 cells. So class 1 present to CD8. It is just like home ministry where the CD8 binds with it, but CD4 circulate in our blood circulation. Now CD4, CD after combining with CD4 cells, now it is naive T cell, it decides how it will progress, like whether it will go to Th1 pathway, Th2 or Th17 pathway. So this is the cellular process which goes on. Now these protein could be viral protein, bacteria, which is exogenous pathway, or they are the cancerous protein, which are processed in this manner. So this is the significance of our MHC complex. Over to Dr. Chakravarti.
I will just add something over here. You have beautifully talked about this very complex subject. What I would also show in this diagram is, see the binding of the peptides. I mean, what we have to realize is that this, the whole concept of MHC or HLA immunology rests on these peptides and how we understand these peptides in terms of T cells. If you look at the MHC class 1, which is on the left, which is bound by CD8, you will see that they are very focused or concentrated. These are monomeric peptides, means they have 9 amino acids on an average. They can be between 8 to 10, but most of them are 9 amino acids. And they fit in like a plug in a socket. They are very much concentrated within the alpha 1 and alpha 2 chains within the groove. But when you look on the right side, the CD4 is binding the MHC class 2. These peptides are much longer, and they can vary anywhere between 15 to 20 amino acids. And at the same time, can you please keep the screen sharing on? Sorry, the CD4 binds to the class 2 peptides, and class 2 peptides are a bit like attached to these groups like Velcro. If that was a plug and socket, it's like a Velcro. And there are particular conserved regions in these peptides which are recognized as epitopes, whether it's for B cells or for T cells. And that will come at a subsequent discussion, and we will have to discuss, at a much greater length at some other time, but that's the basic concept.